Uh, First, as we get into the image software, like this is how the window looks like. Small tab with inclusive of many tools here. And
Yes.
yeah, first thing we have to just click on the file, then click on open.
Hmm.
Then yeah, as we have downloaded the images which sir has posted it in Drive. First, we have to download it from there. Then it will be available wherever you have stored in your computer. Then just go into that folder. Yeah, yeah. This is Saline Control CA1. Like, let me take a first image and show how can we analyze data using image software. Yeah, this is how the image it opens side by side. Here you can as well as see the tool also, image software tool, as well as the picture, the image which we want to quantify. So now just go into the image. Here you can see image option, third one. And here just click type. In that select 8-bit. After doing it, next you'll find option called process. Just click on to that. And next from down the second one, you'll find subtract background. Just click on to that. And yeah, just select light background. Sometimes by default it will show as it is, like background 50 pixels, something like that. Just you have to click OK. Yeah, now your image it turns into like black and white. You can see here there's a difference. First, go into file, open your image which you want to quantify. Then next go into type in the section of image, then select 8-bit. After that, next option, go into process. In process, you have to subtract the background. After doing that, again you go back to image, then click on adjust. In adjust, you will find various options. Go to the fourth option called threshold. You can see it here, threshold. Just click on to the threshold, a small tab will open and you will be able to see these options like minimum and maximum values. It is from this particular image, CA1 control, saline control image, it is 0 and 185. Minimum is 0, you can see here, maximum is 185. Yeah, this is the threshold values. The thing what we have to do is you have to remember these things, minimum and maximum values, that is 0 and 185, so that you have to uh, put the same values, maintain the same values for rest of all the images in the treatment groups and all images which you're going to quantify. You have to maintain the same threshold values for all the images. And here by default, yeah, it will show default. We have to just keep it as such default. And here you will find red, black and white, and over and under, like these three drop downs. From these drop downs, select black and white. If it is already black and white, then you don't have to do anything. Then just exit from this box, just click cross, come out of it. Yeah. After doing that, after checking, putting up the threshold values and all that, then again, yeah, what you have to do is now you can see like plus mark running over your image. When you put the cursor on the image, you can see a plus mark, this plus mark. So just choose small areas, bits and parts of areas where you can find the intensity, like expression of cell, CFOS expression, like black spots you can see here and there. Just those small areas, you choose a little bit of small area using that plus symbol. Like this, it will turn into a square box. You can adjust this also in this way. You like how much ever area you want to measure, you can just adjust it by putting the cursor over it, rolling over it and clicking it. So after choosing this small portion of from the image, then again go to the tool of image J, click on
Okay.
analyze.
Stop here, Alani.
Yes sir.
Little bit of modification. Little bit,
Okay,
please.
okay
And
sir, yes sir.
so here, actually, how that actually rectangle selection is coming. No, no, you keep that. Hello.
Okay, okay sir, okay sir,
Yeah, I'm
I'm
telling you have to do only. I'm telling what actually you missed. So you find what you missed. I'm telling you here, when you are actually telling that that cursor is actually coming as a plus sign,
Yes, sir.
it is because from the tab you have chosen that I tool, that is rectangle selection tool. Can you see that? See here
Uh, this
on the
hands
right.
one, this one,
No,
sir.
no, no,
Scroll.
no, not that one. No, no, left, left. First one, first below the file,
Okay,
that is
sir.
selected. Yeah, that is the rectangle selection option.
Okay, sir.
When you, that's why it is actually allowing you to select. Okay, but
Okay.
here you, instead of actually selecting the design, your choice locations, actually you supposed to select the whole image. Can you select the whole image from top to bottom? Right, top left to bottom left. Right.
Okay, sir.
Yeah. So this is how you have to select. If even the whole, you left little bit up. So maximum you have to from the, because all the images posted here are of same size.
Yes, sir.
Okay. That's why you need not to worry about that. That the extra information will come. It will bring all the information similar because the size is same for all images.
Okay, sir.
Because while taking the images that was set, है ना?
Yes,
So
sir.
you
Okay.
have to select the whole thing, and now you do rest what you did. Okay,
Okay,
guys.
sir.
Hello, guys. All of you. And yeah, just modification a little bit. Instead of selecting the black areas, dotted areas where you can see visually, we ask the computer to select. You consider itself. Computer will see where the black dots are there. Okay, Alani.
Yes, sir. Okay, sir.
Yeah,
I think I have made a mistake in this section, sir. Thank you, sir.
yeah. You have to improve that. Okay. Yeah.
Okay,
Then
sir.
you
Yeah.
you again you modify your results. Yes.
Okay, sir. Okay, sir. I'll do it, sir.
Please continue. Yeah.
Okay, sir. Yeah. Like as in the section under file, you'll find a rectangular box. So with that, you can choose total image as sir mentioned. Like we have to choose the total image and then
Mhm.
go into analyze. Click on to the
Mhm.
measure. So you
Yes.
will get. Yeah. Before doing that, I'm sorry. Uh,
Yeah. Don't save. Yes.
okay. Yeah. In the analyze section, you have to set the measurements. In analyze
Right.
section, you go into set measurements. You have to choose what all the parameters you want to measure. The system you which you want to ask the system to measure in this particular image. Like we have to go click on, just check these check boxes which are mentioned here, whichever parameters you want to include in your data results. Just go into that and click the check boxes. Like area we need to measure, then mean gray value, then here you find integrated density. Just click on to that checkbox, limit to threshold values, and then display label. After checking onto these check boxes, then click OK. So your results, you'll get those values in your result section, like whichever desired results which you want to measure, analyze, that we have choose now. Going into analyze section and setting up the measurements. After doing this, yeah, we have to select the entire picture using this rectangular tab, whole image, then go into analyze, click measure. Then you will get one more tab called result. You can see it here, small tab. And yeah, here we can see like first one, saline control, the name of the label image which you are measuring,
Yeah.
then area. Then you'll, because we have clicked on to the area, we wanted to know those values also. So area we are able to see here, then mean we are able to see, integrated density, that is the actual thing which we want to measure for the expression of CFOS, that, then raw integrated density, minimum and maximum threshold. As I said, like we have to remember this, like minimum is 0 and 195 for our image, because we have to maintain the same minimum and maximum threshold values for rest of the images which you are going to measure further. So this is
Yeah.
how we'll, yes, sir.
Can you do next one? Another one?
Okay, sir. Like now we have to go into file. This result we have to save it. And this
No,
image, this,
we will save together. No, it's all right.
okay,
Alani, we
sir.
can save together. We want to see another one.
Okay, sir.
Whether the area remains the same or it changes a lot. We'll see.
Okay, sir. Now I can cross this, right, sir?
Yeah. Don't
Yeah.
save.
Okay. Then
Don't
again,
save the images because it will change the whatever the processes you have done. The
Yes.
processing, because it's basically a software which allows you to process the image. Okay, so you need not to save them. If you save them, then the image will change.
Okay, sir. Then in the
Please.
same procedure, like yeah, in the
Okay,
same
ma'am.
way, like how we have followed for previously, just click onto the file, go to the open, then choose the next image. Like we are going for next image. In the line, we will take one more control 2 image. Click onto that particular image which you want to measure next. Then click open. Again, this image comes over here. After doing that, same thing, go to the image section. Once the image opens, go to the image section, click on to the type, select 8-bit. After doing this, go to process. In that, you have to subtract the background. Just click OK. It will turn into black and like bright black and white image like after subtracting the background. Then again, go into image section, adjust the threshold because as yeah, get into the threshold in the adjust section. One more pop-up box will open. Here the threshold is already it's the same of the previous image like 0 and 185, so we don't need to modify the threshold or set the threshold. If this varies, we have to set. Just here you can see a option called set. We have to click it and we can set from here, like we can modify here like whichever threshold we want. In this case, our threshold we need it to be 0 and 185. Minimum is 0, up lower threshold, upper threshold is 185. So it is already the same thing as of our first image, so we don't have to change anything. So just click cross, and it is also same, so we don't have to change anything. Just click onto the cross. After doing that, then again go to analyze, just check the setting measurements like whatever we have set is proper. Again, just to make it sure that we are going in choosing the right things like area, gray value, integrated density, and so on. Then again click OK, come back. Now the same process, as we have already choose the rectangular box under the file. Now the plus cursor you'll able to see it on the image when you put it. So just click, and then you'll get this like yellow color box. Drag it, choose the whole image. Yeah, like this, choose the whole image. Yeah, after doing it, go to analyze, click onto the measure. See, we are getting one more value of our second image, that is saline
Yeah,
control 2.
good job. Can you do another one from another group so
Okay,
that
sir.
we can see the threshold changes?
Okay, sir. Okay, I'm going to KA group. I'm taking first image. Caesar's group, first image. KA, Caesar 1, first image. Then click image, then type, then 8-bit, then process, then subtract background, click OK. Then again go to image, adjust the threshold. See, here the threshold value has changed for this image. It is 0 and 234. 234 you can see, and we have to maintain the threshold same for all the images. So now we have to change this. For setting this up, we have to click set. Here you can see an option called set. Just click, one more small pop-up box will come. 0 we want, that is there. Lower threshold, it is perfect. Upper threshold we have to change to 185. 185. Just click one, type 185, click OK. And yeah, now automatically the image is set to the threshold of 0 and 185. Then close this box, go to analyze section, click on to set measurements. Yeah, it is perfect, like whatever we have chosen it is displayed. Then come out of it. Yeah, as put the cursor on your image, this plus sign you'll able to see. Click somewhere, anywhere. Yeah, you'll get this rectangular box. Drag it completely, choosing the full image. Yeah. In this way, we have to select the entire image with that yellow box, rectangular box. After doing it, Go to analyze, click onto the measure. See, third value, KA Caesar, we got it here. Area is this 1335. Mean value we are getting here. Integrated density value. All the values.
Yeah,
Yeah.
one thing here you can see that integrated density by how much? Can you read?
Yes, sir. 214613, sir.
Double the
214. Double lot of control value, sir.
kind of, okay.
Yes, sir.
Here you can see that area changed a bit.
Yes,
You
sir.
know, you are taking a little bit more area as compared to.
Okay, okay,
Yeah,
sir.
so why, Sunil, why you asking why threshold is remain same? Because this actually computer is actually subtracting background, and also it wants to keep how much actually it can consider it as a CFOS expression. Okay, we are telling that zero means no, nothing is there. When there is no dots, then it is zero. But it should not be too much. Actually, if you not keep constant, then it will take unwanted artifacts as a count as well. For example, if you will see this image, there were some artifacts. If you clearly see, one thing I will show you how a CA1 looks like. I'll just share one file. Can anybody open that first thing? Okay, let me see whether that I have posted here on the. Do you know how CA1 looks like? Hello, Sunil. Hello.
Yes, sir.
You know how CA1 looks like? Cornu Amonis one layer.
यस सर देखा था
देखा है? तो मैं उसको एक्चुअली और एक बार दिखा देता हूँ आप लोगों को. नहीं तो क्या होगा, आप if you look at that, then there may be several artifacts you don't want to take into consideration. If there is too much, okay, then it will over predict. Are you getting? Hello,
यस सर
Sunil. See, when you are processing the sections, well, live sections actually you are taking and then you are fixing, then you are putting antibodies. So there may be some artifacts which may be darker than what is CA1 is. Okay, so
यस
what we can do is, if you are considering the first one, that saline images as your normal one, then you keep that highest threshold as it is, so that artifacts can be minimized. By this you may argue that sir, original things may be also hidden or wiped out, है ना? ऐसा
ओके
बोल
सर
सकते हो. आप थोड़ा ध्यान से सुन पा रहे हो क्या मेरा बात?
यस सर
See, if you are taking the saline images as your reference, then you know that there is only a little bit of CFOS expression, and during that you are putting the threshold to such a number, okay, higher threshold, so that only it can show you a required CFOS expression. No much artifacts, because you are not seeing much actual artifacts in that, in the saline one. But when you are actually giving any drug, that may activate also certain other areas where you that may actually what you can say is interfere in your calculation. So you keep that higher threshold also to the same value in all other images where you may say see some artifacts because of interaction with some other cells also, other tissues. So you keep the threshold same. है ना?
ओके सर
So you may lose some data from the treatment, other interventions, but it will be equal loss everywhere. But if you won't actually consider that, then actually that will be biased. सुन पा रहे हो?
यस सर
हाँ. ऐसा होता है ना, जैसे फॉर एग्जाम्पल we want to select some people for a particular job, okay, and we fix the height of the individual should be between 160 to 200 centimeter. सुन पा रहे हो?
यस सर बट सर एक्चुअली अपन जो ये कर रहे हैं तो सर उसमें अपने सर कहीं ड्रग का तो कुछ रोल दिखा रहे हैं
कैसे नहीं दिखा? नहीं दिखा?
हाँ बट सर एक्चुअली अपन ने ये लिया नहीं है ना कि ये ड्रग यूज कर रहे हैं।
तो किया है ना हमने। थर्ड ग्रुप जो है दैट वाज ड्रग, के प्लस ड्रग। आपने पूरा देखा नहीं सकते। saline control is the group where we have got 5 animals. They were given only saline of what? Saline plus saline. But in the second group, that was given saline plus kynic acid in saline. In the third one, kynic acid in saline plus drug in saline.
Oh sir, काइनिक एसिड इज ए ड्रग।
No, that is the inducer. That induces seizure in animals. That actually causes asynchronous firing of neurons because that's a neurotoxin.
ओके सर।
Kynic acid is a neurotoxin that is used in this particular study as an inducer to induce a model, seizure model. सुन पा रहे हो? ये भ्रमित आदमी का रिसर्च है भाई, ये कोई अच्छे मतलब समझदार आदमी का रिसर्च थोड़ी ना है। It's, you know, confused human do like this. They induce disease and then they test the drug. सुन पा रहे हो?
सर
हेलो सुनील जी,
हाँ सर समझ आया।
समझ गए? हेलो,
यस सर।
क्या समझे बताओ? क्या क्वेश्चन था और क्या समझे आप, मुझे बताओ प्लीज।
सर एक्चुअली जो थ्रेशोल्ड है सर वो अपन मतलब कि अपन को सेम रखना पड़ेगा क्योंकि जो
हाँ
क्योंकि सर जो ड्रग है वो अपना मतलब कि सर अपन जो एरिया ले रहे हैं वो मतलब कि अपना जो ड्रग है वो मतलब कि पूरे उस एरिया को अफेक्ट करता है। तो सर अपन को मतलब कि थ्रेशोल्ड चेंज नहीं कर सकते अपन।
ये थ्रेशोल्ड फॉर व्हाट? थ्रेशोल्ड टू डिटेक्ट मैन, थ्रेशोल्ड टू डिटेक्ट एनी ब्लैक एरिया। सुन रहे हो? If black area dots are more than 185, it won't actually consider them. Are you getting?
yes
मैंने
sir,
बताया ना,
detect
जैसे आपको कोई सिलेक्ट करना है किसी एक बंदे को आपके काम के लिए, आपने फिक्स किया, यू फिक्स द हाइट ऑफ द पर्सन शुड बी बिटवीन 160 and 200. So it is basically what it does basically is it actually makes the more than 200 centimeter guy redundant, that you don't include, you are not taking that into your consideration, and also the lower ones, है ना? So basically it's a process so that you can avoid unwanted data, है ना? Unwanted data क्या है ना? While doing this kind of immunohistochemistry, there may be what? Hello, सुन पा रहे हो?
yes sir,
बीच बीच में हाँ हाँ बोलो तो मेरे को पता चल जाएगा आप हो। So इसमें क्या हो रहा है कि they are actually using sections of the brain, rat brain, and
yes sir,
putting antibodies on them to make some color, है ना? तो C first takes those antibodies and gives a color. So during that there may be some artifacts because of the presence of the drug or because of presence of kynic acid, है ना? So
yes sir,
we don't want to include those artifacts. So whatever the threshold that was put in the control animals to detect any expression, we are keeping that as such in all other ones, so that we are not going to over predict, है ना, in other ones, because in that in the other ones there is actually too many dots at different locations. We are actually wiping that out. If more than in any area more than 185 are there, then it will not consider them. It will only consider up to 185. More than that it will just don't consider.
okay sir,
तो इससे क्या हो गया कि सिलेक्शन जो है वो प्रोसेस एक अनबायस्ड हो गया कि भाई इतना इतने ही करेंगे हम, इतने के अंदर
पर्टिकुलर
ही रखेंगे,
जी
हाँ रेंज में।
जी
क्योंकि आपने खुद ही देखा, you just saw ना that cyanic acid group animal, in that higher threshold were 2.235. In some locations this is too dark. सुन पा रहे हो?
Yes, sir.
In some locations it is too dark, so it may be biased one, so that's why we have to fix that 185. Okay ना?
Yes, sir.
चलिए,
जी, थैंक यू सो मच सर। सर देखना पड़ेगा एक्चुअली इसको।
और व्हाट इज 8 बिट? 8 बिट इमेज का मतलब क्या होता है? 16 बिट इमेज का क्या मतलब होता है? व्हाट इज मीनिंग ऑफ 8 बिट 16 बिट इमेजेस? हम्म 2 टू द पावर 8 क्या होता है? 2 टू द पावर 8 कोई बता सकता है?
सर 2 को 8 बार मल्टीप्लाई करना होगा।
करो, बताओ।
सर 256
Right. It is basically 256 colors can be saved in that particular image. You are actually shrinking, or you are actually restricting 256 colors. You may ask, sir, how come there will be so many colors? Actually, because when you are looking at, because even I cannot see every color. There are so many actually things that you cannot see. है ना? सो वो कंप्यूटर में क्या होता है, वो सारे चीज को थोड़ा सा ही डिफरेंस होता है, वो बदल के वेवलेंथ डिफरेंस होता है तो उसको दूसरे कलर मानता है. If there is a little bit difference, then it will see difference in wavelength, it will be a different color. Okay, so in this particular image, if you are fixing 256 colors maximum, then in all images you are fixing it, so there is no bias of colors. Are you guys getting?
Yes, sir.
Hello. So basically, whatever the processing we are doing, all the tools that we are using, that actually minimizes bias. Background we are subtracting because every image is differently processed, so there may be some background which is which need to be normalized. That's why we are subtracting the background. Getting, guys?
Yes, sir.
Yes, sir.
CA1 कैसे दिखाऊं मैं आपको? Only my computer has stopped taking internet from my mobile. Let me select again. तो 16 बिट इमेज में कितना कलर हो सकता है? हम्म 2 to the power 16. हाँ, उस आपने देखा होगा. Yes, 6500. 65K you mean, or what?
Yes, sir.
Hmm?
Yes, sir.
So as you go on increasing the image bit type, so it will increase the size of the file as well. है ना? So that makes this processing very slower. So we have to keep it very actually at the minimum, so that we can, because in this particular image we are specifically interested in one color, so we need not to worry about that, है ना? So we have to keep it at the minimum. Okay, Nayak. This is I'll show you how CA1 looks like. Just for a minute. हेलो गाइस, आप मुझे देख सकते हो?
Yes, sir.
हाँ हाँ, मेरा स्क्रीन देख सकते हो ना?
No, sir.
ओह, मैं आपको दिखाऊंगा। हाँ, okay, here it is. By the time it downloads, I'll go to research design. I have shared this research design presentation already since several days, so you all should go and see it. क्या सोच रहे हो? देखना चाहिए कि नहीं देखना चाहिए आप लोगों को? लेकिन आप देखते हो क्या? इसलिए बहुत मुश्किल है। आप लोग क्या है कि कुछ पकड़ नहीं पाओगे। अचानक मैं कुछ बोलूंगा तो आपको कुछ दिखेगा ही ना। इफ यू विल जस्ट गो थ्रू दैट, जस्ट क्या 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 है उसमें, तो फिर आप लोगों को पकड़ में आएगा व्हाट आई एम सेइंग। इफ यू गो थ्रू दैट, एक्चुअली जस्ट ओवरलुक दैट what are the contents and what sir is going to talk about today, so you can follow me very well. Yeah, it is there. So this thesis has got some images. Okay guys, can you see this image?
Yes, sir.
हाँ, थोड़ा सा करूंगा, थोड़ा नहीं पूरा कर दूंगा। हाँ? देखो, this is one of the section of the rat hippocampus. Rat hippocampus is a banana like structure. But if you look at that, it looks like a slender structure, and it's like a banana. If you cut banana, you'll see the seeds are arranged. Every slice will have the arrangement of the seeds the same in all. Am I right?
Yes, sir.
Yeah, and they are intact. All the seeds are intact. ऐसा होता है कि नहीं होता है?
Yes, sir. Sir, are you talking about TS or LS?
It is TS.
Okay, okay.
Whatever you can see at the moment, you just add on like this. Okay, this image what you are looking at, you just add the slices like this, so it will be a long structure, है ना, like a banana. जैसे आप बनाना काटते हो तो ऐसे सामने दिखता है, ठीक है? You see there are actually seeds arranged, है ना, in a banana slice. आपने देखा है?
Yes, sir.
खाली खाली खा लेते हो, देखते ही नहीं हो लग रहा है ना? तो ऐसा ही इसको भी
Sir,
ऐसे
देख
काटा गया है।
रखा है।
बनाना को आप सीधा ऐसे बनाना रख दो और उसको स्लाइस करते जाओ। इफ यू लुक एट दैट, देन इट विल लुक लाइक दिस इन hippocampal slices. And most interestingly, all the cells remain intact. in every slice. So it starts from here, it takes information from layer 3 of cortex through a perforant pathway. You can see this is an axon. Axon is actually putting the information to or giving input to the dendrites of what? This is dentate gyrus cells. Dentate gyrus आपने देखा है कि नहीं? Did you see dentate gyrus? Hello.
नो सर.
Arey guys, I did you just analyzed? If you have not analyzed, you just go through that. The dentate gyrus cells are nothing but they are called as granular cells. ये जो dot दिख रहा है dentate gyrus में, और if you look at these dendrites of this particular granular cells, this granular cell you will actually, I will teach you all this actually in next course, probably next year, next semester. Okay, I will just actually conclude here saying that these cells you look at here, you are actually quantifying C4 expression is one of the slice of CA1. These are the cells of CA1. Can you see here dotted line? And these are the triangles. They look like a pyramidal cells. They are called as pyramidal cells. In fact, CA1 cells are called as
Pyramidal cells.
pyramidal neurons. So they look like these pyramids. And we have taken a slice of that, and we have actually made an image to analyze CFOS. Similarly, if you look at this one here, this dentate gyrus has been taken as an image to estimate CFOS expression in dentate gyrus, and specifically dentate gyrus ग्रैन्युलर सेल्स डक माउथ की तरह दिखता है, एक डक की तरह दिख रहा है कि नहीं दिख रहा है? देखो मुंह उसका, हिलस जहां पर लिखा है हिलस. हेलो.
यस सर.
Yes, sir.
मेरा कर्सर उधर घूम रहा है, देखो ये हिलस, देखो डक मुंह की तरह दिखता है, मैं इसको डक माउथ बोलता हूँ. इसके बारे में और जानना है तो इसको डाउनलोड करके फ्री तो मिलता ही है, आपको जानना है तो देख लेना, इसमें बहुत सारी चीजें लिखा हुआ है. And but anyhow, we are going to learn this in next semester. For M.Pharm student, I am telling. Okay, so where I was, I wanted to tell you next thing that was, है ना? इतना आपको कोई क्वेश्चन है? Any questions regarding image now? You may have many questions, but if you want to ask now, you can ask, or you can keep it for later. If you can write, or you can email me so that I can call you and give you the answers.
Sir, after we have done the measurements of all the images, then we are going to save it at one place?
Yeah, how to save that? Alani did not show. Alani, you missed that.
Yes, sir. Like at the end, we wanted to save all the data together. So
कैसे करना है? You have to make an Excel sheet. है ना? You have to save as an Excel. Can you show? Did you close that?
no,
Alani,
sir. No, sir. I didn't close it, sir.
do you have that results?
Yes, sir. I do have.
Yeah,
Sir.
can you share them quickly, please?
Yes, sir. Sure.
Because these guys, research scholars, they have done some kind of research in masters, so that's why they have quite quick graphing power. Because only humans can only imagine things what they have already been told. That's very important thing you have to keep in mind. That's why you need not to worry about, because you need to see something so that you can imagine. So Somali, very good question. We'll just show you how you can do it. Yeah, can you
Yes, sir.
maximize the results? Okay,
Sure.
maximize that. Yes, please.
Yes, sir.
Yeah. Now go to file, save as. Yeah, can you from all files you choose Excel, save as types at the bottom.
Sir, it is showing all files only, sir. No drop downs.
Okay,
It will
now from the
automatically
top,
save as. Okay.
Excel.
Sir, I think it's
Okay,
automatically saves as
have
Excel, sir.
as. Okay, so you set the path desktop. Okay.
Okay, desktop. Okay, sir.
Yeah, save. Now go to desktop and see whether it is saved.
Yes, sir.
Directly
Here.
you can go from here. Okay, so from here you can go to Yeah, can you open that?
Yes, sir.
See, Somali, can
Yes, sir.
you increase the size a little bit, Alani?
Yes, sir. Sir,
Yeah,
is it okay?
Somali. Yeah, Somali. When you do that labels, you can see, display label. If you are selected in the measurement, set measurement, then you can see at the B column, you will find 5 saline controls, 5 KS scissors, and 5. Somali, are you listening?
Yes, sir.
Yeah, then you follow what I am telling.
Yes, sir.
What will be the next 5? Here in the
So
labels, you will see
your 5 saline images, 5K scissors, and the 5 other things.
guy key, है ना? And then
Yeah.
area for those 15 of those, and then mean and
Yes,
integrated
sir. All
density.
the
And
results will be in one Excel sheet.
yeah, that's right. And then you delete the other ones. Okay. Only thing is to for confirmation whether you did it correctly or not, to check whether the threshold, minimum threshold and maximum threshold was same for all. To do that, you have to do what?
Take
You
five
have to,
measurements for each.
yeah, no, you have to keep these minimum and maximum to only to verify whether you did correctly. है
Okay,
ना?
sir.
Then you
Yes.
can remove this road integrated density also. You can remove FGH, you can delete. Can you delete quickly? FGH, that is just select FGH.
Sir, I've
No,
deleted.
but delete mean and area also. Yeah, है ना? कॉलम डिलीट करो, आप डिलीट नहीं कर रहे कॉलम्स को. हाँ,
Okay, sir. Once.
yeah, delete columns. Yeah, right click on columns. ऐसा नहीं करते हम we select the C and D. We select only C. Hover your mouse over C and D and keep your cursor on C or D, and then
So it's getting selected entire.
right click. No,
No, sir.
C के ऊपर click करो. C, only on the C. Right click.
Yes.
Right click. Right click on C. Yeah. Then there is delete cells.
Yes.
है ना? Then F4 दबाओ तो फिर दूसरा भी delete होगा. Then you
Yes.
have got labels. Can you stretch B? Can you stretch column B so that you can see the labels clearly?
Yes, sir. I've done.
Okay. After doing this, what you have to do is you have to open one Prism file. Somali, are you following?
Yes, sir.
ये hide करो ना, hide. ये जो
Okay, sir.
stop sharing. Yeah. Go to file.
Sir, it's getting open. Once.
Okay.
Yes, sir.
Right. You make it group, column, sorry, column graphs. है ना?
Yes,
We
sir.
are
Column.
interested in
This
column
fourth.
graphs. Select, yeah. And then say mean and SD करो. Mean and SD.
Yes, sir. Mean with SD.
है ना?
Yes, sir. Create.
Then say okay. Create, please.
Okay, sir.
Now, Somali, Alani, you name them. Actually, first one is column title. You give saline quickly as saline, right?
Yes, sir.
Saline.
Done, sir.
Next,
Integrated density.
controls. K controls.
Yes, sir.
Then third one.
GYK.
Yeah. But then now you type for one. You type just randomly. You type 10, 20, 30, again 10.
Yes, sir.
5 नंबर डालना है आपको. 30, 20, 10 ऐसा लिख दो फिर. हाँ, वैसे 3 3 लिख दो. ठीक है, बढ़िया. इसमें ज्यादा लिखो ना थोड़ा. हाँ, के में बढ़ना चाहिए ना. 50, 50, 60, 70 ऐसा.
Okay, sir.
हाँ, ठीक है ना, बड़ा थोड़ा. हाँ, If we expect drug treatment should have less, so you should type maybe 25, 30, 40.
Yes, sir.
Now you go to analyze.
So, analyze?
Analyze.
Yes, sir.
We will
One
do
way
what analysis? One way ANOVA.
Okay, sir.
Okay. Yeah, post hoc test, you keep it as SNK test, SNK, multiple
SNK.
comparison. Yeah, Newman-Kules, student Newman-Kules.
Okay, sir.
Say okay.
So this is the result, sir.
Yeah, so there is no statistically significant difference that we can see because we have too much sample, all of them. If you
Yes.
go to the data one graphs, you can see there is huge difference in the standard
Error.
deviation. Can you see the bars, error bar?
Yes.
It's
Yes, sir.
too much.
Right.
That's why it is not statistical significance. If you just make them closer, if they were closer, then it would have been statistically significant. Okay.
Okay, sir.
So can you now title कैसे लगाते हैं, कैसे कर सकते हो आप? Yeah, type there. Yeah, just integrated density. Integrated. Is it showing integrated?
Yes, sir.
There is spelling mistake.
Yes, sir.
हाँ, ठीक है. ये treatment groups. Data one on top, because you are going to put no, you need not to do that. No, that's not
No.
needed.
Okay,
No,
sir.
because you are going to have legend where it will explain what this graph is.
Okay, sir.
So you need not to do that. This is what actually your graph should look like, and you put stars if there is statistically significant difference. You have to put stars, and in the legend you have to explain what the star means. And to
Oh,
have some idea about that, you have to go through the Galton et al. Paper. Okay, guys.
okay, sir.
Do you think it is enough, Kamali?
So, so the data we have put in the columns like 10, 20, 50, what all
Sorry.
are those? So the data we have put in the columns now, like
Uh-huh.
10, 20, 50.
Uh,
So what are we supposed to
you
put
bring.
there?
Oh, you are. Why did not we did not show you from the results that actually we downloaded? Can you go to that Excel sheet, Alani? We supposed to copy from there only. Yeah, you
Okay,
have to
sir.
copy the integrated density.
Okay,
Kamali,
sir.
you have to copy the integrated density of those 5 animals of saline
Okay.
control and
Okay.
paste it here. Paste it in the saline control.
Okay. Okay, sir.
That way. Here you can see.
Yeah, yeah. Then
Okay.
again, those 5 of the KS is your
Yes,
control.
Caesar,
Paste.
and have the GYQ there.
Yeah, yeah. So you have to paste, then you can see everything like this. And I would suggest all the master students to keep in touch with PhD scholars, because they have a kind of understanding which connects with me a little bit. So they can give you more information than what you have. Okay. By that you can develop a relationship, brother sister relationship, brother brother relationship, which can actually give you some understanding, because relationship is only for understanding, not
So
for body requirements.
after we have the bar chart, we have to paste it on the doc.
Of course, you have to create that guideline is already given in that course manual. You have to follow that, and you have to make a report
Okay, sir.
according to guidelines. Somali, did not you go through that course manual?
Yes, sir. That's why I'm asking.
You did not go, na?
I did, sir.
You did? Okay, that's
Yeah.
good. Then you might have started working on that then.
Yes, sir. I was confused regarding some things which I have see today. Well, just I think I'm gonna submit it.
You have to copy. How to copy that graph? Can you show? Yeah, how to save an image?
Yes, sir.
Data. Go to data one.
Okay.
Data one. The graphs. Yeah.
Yes, sir.
Can you go to file and then export?
File in export.
Export
Yes, sir.
JPEG. Make a JPEG file. It is smaller, okay, as compared to other image formats. JPG.
JPG.
Keep it 600 dpi. It's at least 600 dpi.
Resolution, sir.
Yeah, right.
600. Okay, sir.
And then choose the path where you want to have this at the bottom. Second path, browse, and you set the path where, because you are preparing your term paper, so your term paper one might have a folder, so you can put this in the folder as a figure one, and
Okay,
figure
sir.
two, like that. What it is saying?
Yes, sir. I have typed figure 1 as a name, file
Ah,
name.
ah,
It is desktop. I'm going to save it, sir.
yes.
And okay.
Okay.
Yes, sir. It is
Go to
saved.
desktop. Go to desktop.
Yes, sir. Here it is.
Open it. Double click the image. Yeah. So here, from here, I think we have already talked about it. Actually, how to make your, how to prepare your report on Word already been explained. Am I right,
Yes.
Somali?
Yes, sir.
Yeah. So you can incorporate this. You can insert pictures. This is as a picture, so you can insert it, and you can right click and insert caption, and you can figure one, figure two. You can name it so that you can make this list. है ना? ये
Yes,
आपको
sir.
you might have all these already in your mind. I today I'm not seeing Deepa being more involved or any other students.
Yes, sir.
Are you there
Yes, sir.
following?
Yes, sir. I'll follow.
Okay, but are you following now? You will follow in future. That is all right. But are you following now?
Yes, sir.
Okay, good. What about others? We have so many students here. 20, around 20 people. Abhit, Sid,
Yes, sir, we are following.
Ankit
Yes,
Kumar
sir. follow कर
Malik.
रहे हैं हम.
All of you are all right. Hansal, Nikita,
Yes, sir.
Yes, sir.
Yes, sir.
Suraj, Nipul,
Yes, sir.
Omkar, Rajesh, Sangam. Sangam, are you there?
Yes, sir.
Sangam is not talking to me much more, so I don't know why.
No, sir. It's not that.
Eh?
अरे ऐसी कोई बात नहीं है, सर बात कर रहा हूँ.
है ना? समझ रहे हो? मैं थोड़ा थोड़ा कड़क कुछ बोलता हूँ तो आपको खराब लगता है क्या बहुत?
नहीं सर, बिल्कुल भी नहीं सर. आपका काम आप बहुत अच्छे से कर रहे हो.
है ना? ऐसा लगता है आपको?
हाँ सर, आपकी ड्यूटी है. थोड़ा बहुत गलत भी बोल दोगे तो हमारे अच्छे के लिए ही तो है.
गलत कभी अच्छे के लिए हो सकता है?
मतलब सर आप कड़क बोल रहे हो ना, तो उस रिस्पेक्ट में कह रहा हूँ मैं.
उसलिए मैं गलत नहीं बोला उसको, है ना? कड़क बोलना
हाँ
थोड़ी ना गलत।
मतलब सर कड़क वही सर, इसलिए
हाँ,
तो साइंटिफिक नेम बनाए गए हैं. कोई कहीं कुकुर बोलता है, कोई कहीं कुत्ता बोलता है. मतलब मैं गलत बोल रहा हूँ, आप कड़क बोल रहे हो.
नहीं
मतलब
नहीं
भावना समझो ना.
नहीं नहीं, कुत्ता और ये
नहीं
कुकुर थोड़ी
मैं
ना अलग।
कह रहा हूँ मतलब अलग अलग रीजन में एक ही चीज को अलग अलग नाम से बोलते हैं ना, तो मैं वो कह रहा हूँ. आप कड़क बोल रहे हो, मैं उसी चीज को मतलब
नहीं,
मैं.
कड़क का मतलब एक ही होता है. देखो, कड़क मतलब किसी को थोड़ा
हाँ सर, मतलब किसी को ऐसा बोलना जिससे किसी को बुरा लग सकता है. अब
क्यों बुरा
उसके
लग
ऊपर
सकता
है ना,
है?
बुरा लगे ना.
मीनिंग ऐसा नहीं है. इट इज नॉट लाइक दैट. आप
अच्छा.
कड़क जैसे जैसे बताओ, ए पीस ऑफ वुड इज, हिंदी में बोल पूछूं तो एक लकड़ी का जो पीस है वो कड़क होता है या नरम होता है?
सर कड़क या सख्त या हार्ड, मतलब कुछ भी. हाँ.
है ना? तो उसका सही चीज पकड़ो ना यार. नहीं, उसीलिए तो आप लोगों के साथ संवाद नहीं हो पा रहा बढ़िया से. आपने कड़क का मीनिंग
अच्छा
कुछ
अच्छा
और समझ रखा है, जो कि जिस बिल्कुल अस्तित्व में ऐसा कोई मीनिंग ही नहीं है.
अच्छा
कड़क
अच्छा.
मतलब क्या है? जैसे कड़क बोलना मतलब डांट रहा
कड़क
हूं.
बोलना मतलब किसी को डांटना.
हाँ, अभी डांट क्यों रहे? आपको मैं क्यों डांटू बताओ?
सही बात।
क्या दुखी देखने के लिए डांटू मैं आपको?
हम्म,
मैं आपको आप बुरा मानने के लिए डांटू क्या मैं आपको, या आपको कुछ समझाने के लिए डांट रहा हूँ? समझ लो इसको अच्छे। हेलो संगम, गायब हो गए क्या?
यस सर, यस सर।
आप तो ध्यान से सुनो तो कुछ फिर समझ नहीं पाओगे, जिंदगी भर ऐसे ही गधे की तरह रह जाओगे, फिर संगम बोलोगे कि सर क्या यार, सर मानव को गधा बोल रहे हो यार फिर से, है ना? अरे भाई गधा के जैसे ही तो जिंदगी जी रहे हैं हम लोग, कुछ भी मान के बैठे हैं यार।
सही बात है सर।
आप बताओ कड़क बोलना मतलब क्या है? आपको मैं कड़क किस क्यों बोल रहा हूँ बताओ? कड़क मतलब ऐसे मैं आपको है